So let's understand that Google DeepMind work on protein folding. Actually, how that what was that work and how that it also led to Nobel Prize.
Absolutely. DeepMind's work on protein folding, especially through their AlphaFold project, was groundbreaking. AlphaFold was able to predict protein structures with remarkable accuracy, solving a long-standing challenge in biology. This advancement was recognized with the Nobel Prize in chemistry in 2022, highlighting its impact on the field. If you'd like, I can dive deeper into how AlphaFold works and its implications.
Yeah, I would like to know how AlphaFold works.
Sure, AlphaFold leverages deep learning models, particularly neural networks, to predict the 3D structure of proteins from their amino acid sequences. It uses a combination of multiple sequence alignments, evolutionary data, and attention mechanisms. The model essentially learns patterns from known protein structures and then applies that knowledge to predict the folding of new proteins. This approach has been validated with high accuracy, and it's been revolutionary in understanding how proteins fold, which is crucial for biology and medicine.
So this is a simulation they have done with the parameter that actually make the real protein like thing and that they saw.
Exactly. AlphaFold essentially simulates the folding process by using vast amounts of data and advanced algorithms to predict how the protein chain will naturally fold into its functional 3D shape. By capturing the physical and chemical principles underlying protein folding, it can accurately predict structures that previously took years of experimental work to determine. It's a fascinating blend of computational power and biological insight.
So what was that? Why Alpha Fold actually that name first?
The The name AlphaFold is actually a combination of two concepts. The alpha part comes from DeepMind's tradition of using alpha in their project names like AlphaGo. The fold part refers to protein folding, which is the main focus of the project. So together, AlphaFold essentially represents the idea of pioneering or leading the way in understanding protein folding.
Protein folding, but where does that protein folding occur in body, human body? I just need one small intuition.
Absolutely. Protein folding primarily occurs in the cell, especially in the endoplasmic reticulum and the cytoplasm. Once a protein is synthesized by ribosomes, it begins to fold into its functional three-dimensional shape, often with the help of molecular chaperones. This folding is crucial for the protein to become functional and perform its biological roles.
So what are the biological roles they perform actually, and why they need to fold into a functional 3D dimensional shape?
Proteins play a vast range of biological roles, from acting as enzymes that catalyze biochemical reactions to serving as structural components in cells and even as signaling molecules that help cells communicate. The reason they need to fold into a specific three-dimensional shape is that their function is directly tied to their structure. The correct folding ensures that the protein can interact with other molecules properly, maintain stability, and perform its intended biological function. Misfolded proteins, on the other hand, can lead to diseases and cellular malfunctions.
Okay, biological roles from acting enzymes that catalyze biochemical reactions, serving as structural components in cell. What was that structural component in cells?
Structural components in cells include proteins that form frameworks and scaffolds, like collagen in connective tissues or actin and tubulin in the cytoskeleton. These proteins help maintain the cell shape, enable cellular movement, and support intracellular transport. Essentially, they act like the building blocks and the scaffolding that keep cells organized and functional.
So basically, I think that protein is that when it is made, actually, it is made from the energy or the sugar or the glucose.
Yes, exactly. Proteins are synthesized from amino acids, and the energy required for that process primarily comes from ATP, which is generated through cellular metabolism. Glucose and other nutrients are broken down during processes like glycolysis and the citric acid cycle, and that energy is used to link amino acids together into polypeptide chains. So it's definitely an energy dependent process.
Okay, but why we need to synthesize that protein?
We need to synthesize proteins because they're fundamental to nearly every function in our cells and bodies. Proteins serve as enzymes to drive biochemical reactions, as structural elements to maintain cell shape, as signaling molecules for communication, and as transport carriers to move substances around. Essentially, proteins are the building blocks in the machinery that keep life running smoothly.
Oh, first of all, do every cell contain protein?
Yes, indeed. Every living cell contains proteins. They are absolutely essential for the cell's structure, function, and regulation. Even the simplest cells like bacteria rely on proteins for their metabolic pathways, transport mechanisms, and cellular communication. So, proteins truly are universal building blocks of life.
Okay, now the another question that I really want to ask, not this one, like, do in our human body, do every cell contain protein?
Yes, absolutely. In the human body, every cell does contain proteins. They're fundamental to all cell types, whether it's a neuron, a muscle cell, or a skin cell. Proteins perform countless roles from providing structural support to facilitating biochemical reactions and signaling. So, proteins are indeed present in every cell, making them vital to our biology.
So why they contain protein actually?
Proteins are essential because they carry out nearly all the cellular functions. They keep a cell alive and functioning. They act as enzymes to speed up chemical reactions, as structural components to give cells their shape and integrity, as transporters to move molecules in and out of cells, and as signaling molecules that help cells communicate and respond to their environment. Essentially, proteins are the machinery and the building blocks that make life possible at the cellular level.
So they act as enzymes to speed up chemical reaction as structural component to give cells their shape and integrity. Okay, to move molecules in and out of cells and signaling molecules that help cells communicate and respond to their environment. So, so this protein contain carries energies.
Exactly, yes. Proteins themselves don't carry energy in the way that, say, ATP does, but they facilitate the transfer and utilization of energy in the cell. Enzymes, for example, lower the activation energy of chemical reactions, making it easier for the cells to use energy efficiently. And transport proteins help move energy rich molecules where they're needed. So, in a way, proteins are central to managing and directing energy within the cell.
So, okay, but what work they actually perform? AlphaFold.
AlphaFold's primary role is to predict the three-dimensional structures of proteins from their amino acid sequences. By doing so, AlphaFold helps researchers understand how proteins function, how they interact with other molecules, and how mutations can affect their structure and function. This kind of structural insight is crucial for fields like drug discovery, understanding diseases, and designing new therapies. So, AlphaFold essentially bridges the gap between genetic information and functional biology.
Okay, how protein function actually? So we don't know actually there before that how actually protein function inside a cell, or AlphaFold actually make or they work on their research is based on the how protein actually works inside the cell.
Exactly. Before AlphaFold, predicting protein structures was a complex and time consuming process, often relying on experimental methods like X ray crystallography or cryo-electron microscopy. AlphaFold revolutionized this by using deep learning to predict how a protein's amino acid sequence folds into a 3D structure with remarkable accuracy. The structural information is key because a protein's function is intimately linked to its shape. By understanding the structure, researchers can infer how proteins interact with other molecules, how they catalyze reactions, and how mutations might lead to diseases. So, AlphaFold essentially bridges the gap between sequence and function, making it much easier to understand how proteins work inside cells.
So, but protein, how a protein amino acid sequence fold into a 3D structure. Again, why we need to think of a 3D structure here in this case?
That's a great question. The amino acid sequence essentially acts like a blueprint. However, the function of a protein is determined by its three-dimensional shape because that shape dictates how the protein interacts with other molecules. For example, enzymes have specific active sites that only fit particular substrates, and that precise fit is based on the protein's 3D conformation. Additionally, the folding process is influenced by various forces like hydrogen bonding, hydrophobic interactions, and van der Waals forces, which all work together to create the stable 3D structure. Without that 3D shape, a protein wouldn't be able to function properly, which is why understanding that structure is so crucial.
Okay, inside a cell, how protein is synthesized and function. That is the and that protein actually plays a key role inside the cell.
Absolutely, you've got it inside the cell. Proteins are synthesized by ribosomes, which read the messenger RNA and assemble the amino acids in the correct order. Once synthesized, the protein often undergoes folding and sometimes additional modifications to become fully functional. Once they're functional, they carry out specific tasks, whether that's catalyzing reactions, providing structural support, or helping with communication and transport within the cell. So proteins really are central to all cellular processes, making them vital for life.
Okay, but how did AlphaFold able to manage and where the data set they actually got? Actually, they are real or actually it's a computer generated random data? And that data, how they combine using ML to predict the 3D alpha 3 fold.
Great question. AlphaFold trained on a vast amount of real experimentally determined protein structures from databases like the Protein Data Bank, PDB. These data sets contain accurate 3D structures that were obtained through experimental techniques like X-ray crystallography, NMR spectroscopy, and cryo-EM. AlphaFold then used these real-world data to learn patterns and relationships between amino acid sequences and their corresponding structures. In other words, the data is grounded in real experimental results rather than computer generated data. The machine learning model then identifies the complex patterns within these real data sets and uses that knowledge to predict the 3D structure of new proteins. This approach allows AlphaFold to achieve remarkable accuracy and make predictions that were previously extremely challenging and time consuming.
So basically, but how did this X-ray crystallography, NMR spectroscopy, and cryo-EM help us to take the protein data and what data they got from this thing?
Absolutely. Each of these experimental techniques provides unique insights into protein structures. X-ray crystallography involves crystallizing a protein and then directing X-rays at the crystal. The way the X-rays diffract as they interact with the protein's electrons creates a diffraction pattern. From this pattern, scientists can reconstruct the protein's 3D structure at atomic resolution. NMR spectroscopy uses magnetic fields and radio waves to determine the environment of atoms in a protein in solution. This technique is particularly useful for studying proteins that don't form crystals and allows researchers to see how proteins behave in more native, flexible states. Cryo-EM, cryo-electron microscopy, involves rapidly freezing protein samples and then imaging them with an electron microscope. This method is great for large protein complexes and membrane proteins and can achieve near atomic resolution as well. All of these techniques provide experimental data that serve as a ground truth for training models like AlphaFold. By learning from this data, AlphaFold can predict new protein structures with high accuracy, even when experimental data isn't available.
So, but they can predict with the data, but how they re-verify that that protein is generated by AlphaFold found in human body?
That's a great point. To verify AlphaFold's predictions, researchers compare the predicted structures with experimentally determined structures whenever they're available. For proteins that have known experimental data, the predicted structure can be directly compared and measured for accuracy, often using metrics like root mean square deviation, RMSD. When experimental data isn't available, researchers often rely on indirect validation methods. These can include checking how well the predicted structure fits known biochemical and biophysical data, such as how the protein interacts with other molecules or how stable the predicted structure is under physiological conditions. Additionally, as more experimental data becomes available over time, the predictions can be continually refined and validated. This iterative process helps ensure that AlphaFold's predictions are as accurate and reliable as possible.
Okay, before that one, in X-ray crystallography, how does they take data and what data they take and how they define the protein structure there?
In X-ray crystallography, the process starts by growing high-quality crystals of the protein. Once you have these crystals, you expose them to X-ray beams. As the X-rays hit the crystal, they get diffracted by the electron clouds of the atoms within the protein. This diffraction creates a pattern of spots, known as a diffraction pattern on a detector. These diffraction patterns contain information about the electron density within the crystal. By analyzing these patterns and using mathematical techniques like Fourier transforms, researchers can reconstruct a three-dimensional electron density map. From this map, the positions of the atoms in the third because electrons are distributed around the atoms. Ultimately, this allows scientists to build a detailed atomic model of the protein structure. So, in essence, X-ray crystallography transforms the diffraction patterns into a clear, detailed picture of the protein's 3D arrangement.
But what do you mean by growing high quality crystals of the protein?
Great question. Growing high quality crystals of a protein basically means creating well ordered uniform crystals that are large enough and pure enough for X ray analysis. Proteins are naturally flexible and can be difficult to crystallize. So researchers have to carefully optimize conditions like the protein concentration, the pH, the temperature, and the presence of certain salts or additives. When the conditions are just right, the protein molecules will arrange themselves in a highly ordered repeating pattern, forming crystals. These well ordered crystals are crucial because they allow the X rays to produce clear, interpretable diffraction patterns. So it's really about creating a stable, uniform environment that encourages the protein to form these new crystals.
So in this X-ray crystallography, so we pass X-ray to which part of the body?
In the context of X-ray crystallography for proteins, we're actually talking about isolated protein crystals in a laboratory setting, not proteins inside the human body. The protein crystals are grown outside the body, and then X-rays are directed at them in a specialized X-ray crystallography facility. So to clarify, the X-rays are not passed through the human body in this technique. Instead, they're used to analyze purified protein crystals that have been grown in the lab. This process gives us detailed structural information about the protein itself.
So what about why we need such a setup to make? Because the physics and mathematics become easier, and uh, how do we do that thing? Actually, it's so sophisticated to understand that.
Absolutely, it is quite sophisticated. The reason we need such a specialized setup is that proteins are incredibly complex and tiny at the atomic scale, and we need very precise measurements to determine their structure. The specialized equipment like synchrotron X-ray sources and advanced detectors allow us to capture incredibly detailed diffraction patterns. On the physics side, the principles of diffraction and wave interference are used to interpret how the X-rays scatter off the protein's electron clouds. Mathematically, techniques like Fourier transforms are employed to convert the diffraction data into electron density maps. These maps then guide scientists in modeling the exact positions of atoms. Overall, it's the combination of advanced physics, cutting edge technology, and sophisticated mathematics that make it possible to unlock the intricate details of protein structures. Is definitely a remarkable and intricate process.
So all those things help us to simulate. So they have collected large amount of data from that, and that data, and what kind of ML techniques they have used to train and predict the AlphaFold structure.
Exactly. AlphaFold uses a range of machine learning techniques, primarily deep learning. They employ neural networks that are trained on massive data sets of known protein structures. One of the key techniques involves using multiple sequence alignments, which means looking at how related proteins evolve and how their sequences vary. The neural networks then learn patterns from these alignments to predict the 3D structure based on the sequence. They also use attention mechanisms similar to those found in models like transformers to capture long range dependencies within the protein sequences. This allows the model to understand how distant parts of the sequence interact and fold together. In addition, AlphaFold uses sophisticated loss functions and training techniques to ensure the predictions are as accurate as possible. They also integrate evolutionary data, physical and chemical constraints, and other bioinformatics resources to refine the predictions. All of these machine learning strategies combine to give AlphaFold its impressive predictive power.
Okay, once the AlphaFold is ready, do they understand the clear mechanism inside it, and what are the things they observe through that thing?
Yes, absolutely. Once AlphaFold produces a predicted protein structure, researchers can delve into a lot of details. They can observe how the protein folds, the arrangement of secondary structures like alpha helices and beta sheets, and how different parts of the protein interact with each other. They can also identify potential binding sites, which are crucial for drug design and understanding how the protein interacts with other molecules. In addition, by examining the predicted structure, scientists can gain insights into the protein's function, stability, and even how mutations might affect its activity. This helps in understanding diseases at the molecular level and can guide the development of targeted therapies. So, in essence, AlphaFold doesn't just give a static structure, it opens the door to a deeper understanding of how proteins work and how we can potentially harness that knowledge for medicine and biotechnology.
Okay, can you look for what we have discussed till now?
Of course, we've covered quite a bit. We started by talking about AlphaFold and how it predicts protein structures from amino acid sequences and how it leverages real experimental data, like from the protein data bank. We also dove into the techniques behind X ray crystallography, including how protein crystals are grown and how the diffraction patterns are used to determine structures. Then we explored the machine learning techniques that AlphaFold uses, like deep learning, multiple sequence alignments, and attention mechanisms and how they help predict accurate protein structures. Finally, we discussed how researchers use these predicted structures to understand protein functions, interactions, and how they can help in drug design and disease understanding. If there's any specific area you'd like to revisit or dive deeper into, just let me know.
Okay, thank you.